if you've ever stopped to really look at a flowering plant, an angiosperm, you're seeing, well, an organism that's truly mastered reproduction, like in ways no other plants have.
Mm-hmm.
And that moment, when pollen lands on the stigma, that sticky part, it feels like the end, but it's really just the beginning. It kicks off this incredibly complex, sort of hidden biological race.
Oh, absolutely. It's this whole intricate dance happening completely out of sight. So our goal today really is to unpack that, to explore the sequence inside the flower.
Yeah.
We want to trace it right from the pollen landing, that's the pollen pistil interaction, through the really unique angiosperm thing, double fertilization, and then finish up with what happens after fertilization, how the seed and fruit actually get built.
Right. So for you listening, this is kind of your shortcut to understanding that complex coordination, especially how they manage two separate fertilizations at once, one for the embryo, the future plant,
Which is diploid, or TOM.
and the other for its food supply, the endosperm, which is triploid, 3N.
Exactly. That triploid endosperm is key. It's a defining feature.
Okay, so let's start at the beginning then. The gatekeepers. Pollen landing on the pistil.
Right, the pollen pistil interaction. This covers everything from the moment pollen touches down on the stigma, through its germination, and the pollen tube growing down, all the way until it enters the ovule.
And you called it a dialogue earlier. That seems important. It's not just sticking there. The pistil is actively involved, kind of checking the pollen's ID.
Precisely. It's a chemical dialogue. The pistil has these recognition systems. It has to decide, is this pollen compatible or not? Friend or foe, essentially.
So rejection can happen right there.
Oh, yes. Immediately. If it's accepted, though, the pollen grain hydrates, swells up, and starts to germinate, sending out that pollen tube down through the style.
And how the pollen prepares its cargo, the male gametes, differs a bit, right?
Yeah, there's a slight variation. In what we call two-celled pollen, the generative cell inside divides into the two male gametes while the tube is growing down the style.
Kind of assembling the package en route.
Exactly. But in three-celled pollen, that division has already happened. The pollen grain lands already containing the two male gametes, ready to go, a bit quicker off the mark.
Interesting. Faster delivery. Now, I know researchers study this germination outside the plant sometimes, using something called the hanging drop method.
Ah, yes. It's a classic technique, surprisingly simple, actually. You basically create a little drop of liquid medium, usually about a 10% sugar solution with some key salts, like calcium, boron, and hang it from a cover slip over a depression slide.
So you can watch the pollen germinate under the microscope.
That's the idea. It mimics the nutritional environment of the stigma and style just enough to get the pollen tube to grow where you can observe it easily.
Cool. But back in the actual flower, once that tube is growing down the style, it needs to physically get into the ovule, which is tucked away down there.
Right. And this is where we see some different strategies, some architectural variety in how the tube enters the ovule itself. The most common way, by far, is what's called paragamy. The tube just goes straight in through the little opening that's already there in the ovule wall, the micropyle. Most flowering plants do this.
The front door, basically.
Pretty much. But then you have exceptions that show evolution trying different things. Like in Casuarina, they use chalazogamy. The tube grows all the way down, bypasses the micropyle, goes around to the opposite end of the ovule, the chalaza, and enters there.
Wow, the back door route.
Kinda, yeah. And then there's mesogamy, seen in plants like Cucurbita, the squash family. Here, the tube penetrates through the side walls, the integuments, or even through the stalk, the funicle.
So multiple ways to get into the ovule building. But once inside, doesn't matter which door it used.
That's the fascinating part. No matter how the tube gets into the ovule, the final step entering the actual embryo sac within the ovule is almost always the same. Highly standardized.
Okay, this sounds critical. What's the standard procedure?
It always enters the embryo sac at the micropylar end, always. And specifically, it enters into one of the two synergid cells, which are right next to the egg cell. And that synergid cell then degenerates.
It sacrifices itself. Why?
It seems to be part of a precise guidance system. It's called chemotropic guidance. Those synergid cells release chemical attractants. They have a specialized structure called the filiform apparatus, which looks like a bunch of finger-like projections, perfect for secretion.
Like a chemical beacon.
Exactly, a landing flare. It guides the pollen tube tip right to that spot. And once the tube penetrates the synergid breaks down, maybe helping release the two male gametes right where they need to be, next to the egg and the central cell. It's like a cellular concierge ensuring perfect delivery.
Incredible precision. Now, given how picky the pistil is at the start, How do plant breeders handle this for artificial hybridization if they want to make a specific cross?
Well, they have to intervene manually. They use techniques like emasculation, physically removing the plant's own anthers before they mature so it can't pollinate itself.
Okay, prevent self-pollination.
Right. And then bagging. They cover the emasculated flower, usually with a paper or plastic bag, to prevent any stray, unwanted pollen from landing on the stigma. Then they can manually apply the specific pollen they want.
Controlling the whole process. Makes sense. And something else mentioned was the crowding effect. Pollen works better in groups.
Yeah, that's consistently observed. Pollen germination rates and tube growth are often much better when there's a high density of pollen grains on the stigma. They seem to chemically stimulate each other somehow. Safety in numbers, maybe.
A team effort. Okay, so the tube has arrived, guided perfectly. The male gametes are released. This brings us to the main event, right? The thing only angiosperms do.
Double fertilization, yes. This is the absolute signature move of flowering plants. It involves two separate fusion events happening inside that tiny embryo sac, almost simultaneously.
Two fusions. Okay, let's break them down. Event number 1.
Event 1 is called syngamy, or sometimes generative fertilization. This is the more, let's say, conventional one. One of the male gametes, which is haploid, 1N fuses with the egg cell, also haploid 1N.
And the result is?
The diploid zygote, a 2N. This is the cell that will direct and develop into the embryo, the actual future plant.
Okay, simple enough. 1N plus 1NX2N. Wait. But there were 2 male gametes delivered. What about the second one?
This is where the double part comes in. The second male gamete, also 1N, doesn't fuse with the egg. Instead, it fuses with the secondary nucleus in the center of the embryo sac.
Secondary nucleus. Remind me, where did that come from?
That nucleus is usually already diploid, 2N. It forms earlier when the 2 polar nuclei in the central cell fuse together. So you have 2 1N nuclei fusing to make one 2N nucleus.
Got it. So the second male gamete finds this already 2N nucleus.
And fuses with it. This fusion is called triple fusion, or vegetative fertilization. You're fusing 3 nuclei in total, effectively. The 2 polar nuclei, which already combine, and the second male gamete.
So 1 male gamete plus the 2N secondary nucleus, that makes 3N.
Exactly. The result is the primary endosperm nucleus, or PEN, and it's triploid, 3N. This cell will divide to form the endosperm, that food reserve we talked about.
Wow. Okay, so one fertilization makes the 2N baby plant, the other makes the 3N packed lunch. That's incredibly efficient.
It is. It ensures the plant doesn't invest resources in making food unless fertilization of the egg has actually occurred. If you count them up, it involves 5 nuclei, the egg, the 2 polar nuclei, and the 2 male gametes, and 3 actual gametes participating.
Amazing coordination. But you mentioned life is messy. Are there variations or glitches in this process?
Oh, yes. Sometimes things don't go quite to plan. You can get polyspermy, where more than one pollen tube manages to enter the ovule. That means extra male gametes milling around.
Does that cause problems?
It can, potentially leading to abnormal development. And then there's a really strange one called heterofertilization.
Hetero. Different fertilization. What does it mean?
It means that the 2 fusion events, syngamy and triple fusion, are actually carried out by male gametes that came from 2 different pollen grains, from 2 different pollen tubes that both reach the embryo sac.
Wait, so the embryo could have a different genetic father than the endosperm surrounding it?
In theory, yes. It's been documented in maize, Zea mays. It certainly complicates tracing parentage.
That's wild. Any others?
There's also semiagamy, where a male gamete might enter the egg cell, stimulating some division, but actual nuclear fusion doesn't happen, sort of a failed fertilization attempt.
Okay, but assuming everything goes right, syngamy happens, triple fusion happens, what's next? The building phase.
Exactly. Post-fertilization events. This is where all the structures we recognize start to form. The ovule develops into the seed, the ovary surrounding it develops into the fruit. And inside the developing seed, the zygote grows into the embryo, and the PEN develops into the endosperm.
It's the grand construction project.
It is. And interestingly, in most plants, the zygote actually waits a bit. It usually doesn't start dividing until after the primary endosperm nucleus has divided a few times and started building up that food supply.
Lunch needs to be served before the guest starts eating.
A good way to put it. So let's look at that lunch first. Endosperm development. It comes from that triploid PEN, filling up the central cell, and its whole job is providing nutrition for the embryo.
And it can take very different forms, right? Not always just like flowery stuff.
Oh, absolutely. Huge diversity. It can be primarily starchy, like you find in grains, rice, wheat, corn. It can be proteinaceous. Think of the aileron layer in a maize kernel. That's endosperm packed with protein. It can be very oily, castor bean, coconut.
And you even mentioned stomy.
Yeah, sometimes it becomes incredibly hard, lignified, like in the pit of a date palm or the nut of the betel nut. That hard material is actually endosperm.
Wow. And structurally, how does it form? Are there different ways it develops?
Yes, primarily three types, based on how the cell divisions happen. The most common type in maybe 56% of angiosperm families is nuclear endosperm,
Nuclear.
meaning the PEN undergoes repeated nuclear divisions without forming cell walls right away. So you get this big cell filled with lots of free-floating nuclei in the cytoplasm. Eventually, cell walls might form around them or maybe not.
Ah, like liquid endosperm.
Exactly. The water inside a young coconut, that's free nuclear endosperm, liquid food. Later, as the coconut matures, cell walls form around nuclei near the edge, creating the solid white coconut meat.
Which is cellular endosperm. So coconut has both.
It's a fantastic example of double endosperm development. Starts nuclear, the water, becomes cellular, the flesh. The second main type is cellular endosperm, where cell wall formation happens immediately after each nuclear division. So it's cellular from the start. Think of plants like petunia.
Okay, nuclear or cellular. Any other types?
There's a third, less common type called halobial, which is kind of intermediate, but nuclear and cellular are the big ones. Now, related to endosperm, there's this fascinating effect of the pollen.
Right, you mentioned the pollen's influence can extend beyond just fertilization. Xenia and metacasnia.
Exactly. Xenia is the direct effect of the pollen's genetic makeup on the character of the endosperm itself. Remember, the endosperm is part male gamete derived, 1N, and part maternal, 2N.
So the father's genes can show up in the food.
Precisely. The classic example is maize kernel color. If you pollinate a maize plant that normally has yellow kernels with pollen from a plant with blue kernels, some of the resulting kernels on that same cob might actually be blue. The pollen's genes for color are expressed in the 3N endosperm.
Okay, that makes sense because the endosperm is partly from the pollen, but metaxenia, that does further.
It does. Metaxenia is the effect of the foreign pollen on the maternal tissues of the fruit outside the endosperm. Things like the fruit size, shape, or ripening time.
Wait, how? The fruit tissue is purely maternal, isn't it? 2N from the mother plant.
It is. That's what's so puzzling and fascinating. It suggests that substances produced perhaps by the pollen or by the developing seed under the pollen's influence are somehow affecting the purely maternal fruit tissues developing around it. It was first really studied in date palms, where the type of pollen used to pollinate could affect the size and ripening time of the dates.
So a pollen donor is influencing the mother's body, essentially.
Yeah.
That's quite an...
It really challenges our simple ideas about genetic boundaries. Okay, so we have the endosperm developing. Now, let's look at the main event for the next generation. Embryo development, or embryogeny.
Growing the actual plant from that 2-hampton zygote?
Right. We usually look at two main patterns, dicot and monocot. The classic dicot model is often Capsella, shepherd's purse. The zygote undergoes an unequal division.
Unequal?
Yeah, it divides into a large cell towards the micropyle end, called the suspensor cell, and a smaller cell towards the interior, the embryonal cell.
What does the suspensor do?
The suspensor elongates, forming a stalk that kind of pushes the developing embryo deeper into the nutrient-rich endosperm. It acts like an umbilical cord, helping transport nutrients, and anchors the embryo.
And the small embryonal cell.
That's the one that forms the embryo proper. It divides repeatedly, going through some recognizable stages. First, a sort of undifferentiated ball of cells, the proembryo, then a globular stage.
Okay.
Then, as the cotyledons, the seed leaves, start to develop, it takes on a heart-shaped appearance. Then elongates into a torpedo-shaped stage before reaching the mature embryo form.
And that mature dicot embryo has.
The key parts are the embryonal axis, with the plumule, which will become the shoot at one end, and the radicle, which becomes the root at the other. And attached to this axis are the two cotyledons.
The two seed leaves, characteristic of dicots. Got it. How does the monocot embryo differ? Grasses, grains, palms?
Well, the early divisions can be a bit different, and the suspensor is often just a single cell, or very small. But the big difference is in the cotyledon situation. Monocots, by definition, have only one cotyledon.
Just one.
And in grasses, for example, this Single cotyledon is usually large, shield-shaped, and called the scutellum. It lies sort of laterally against the embryonal axis, but often looks like it's terminal. Its main job is to absorb nutrients from the endosperm.
And monocots often have those protective sheaths, right?
Yes, very important, because they only have that one leaf blade initially pushing up through the soil. The developing shoot tip, the plumule, is covered by a protective cap called the coleoptile.
Coleoptile protects the shoot.
And the developing root tip, the radicle, is covered by a different protective sheath called the coleorrhiza. Think of them as protective helmets for pushing through the soil.
Coleoptile for the shoot, coleorrhiza for the root. Very neat. So we've journeyed right from that pollen grain landing through the chemical checks and balances, the amazing double fertilization, creating both the taken embryo and the three-on food, and finally watched them develop into the basic structure of the seed. It's incredibly complex, but also beautifully logical.
It really is. The whole system is geared towards ensuring successful reproduction, selecting the right partner, securing the food supply before investing in the embryo, and packaging it all up for survival.
And reflecting on that pollen influence, xenia and metaxenia, it leaves you thinking.
Definitely. The idea that genetic material from essentially a visitor, the pollen, doesn't just contribute to the next generation's DNA, but can actively influence the characteristics of the food supply and even the surrounding maternal fruit tissue. Well, it really makes you wonder, how far does that genetic conversation actually reach? How much are the flowers and fruits we see shaped not just by the mother plant, but by the pollen it happened to receive? It's a deeper level of interaction than we might assume.